Movement is an important part of our lives. Not just for walking or picking up objects, it's essential for completing everyday tasks like writing. It helps us interact with our environments and engage socially with other people. But what if simply imagining movement could impact how we move? Motor imagery is when you imagine yourself performing an action without physically doing that movement. It seems important for our ability to plan and control movements within everyday life. So having issues with motor imagery may contribute to movement problems we see in some disorders, like developmental coordination disorder or DCD. People with DCD have difficulties coordinating their movement. This makes it hard for them to complete everyday tasks, like doing up buttons or using utensils when eating food. Unfortunately, although it affects around 300,000 children in Australia alone, we don't know how this disorder develops. Interestingly though, we conducted a review and found people with this disorder also have difficulties imagining movement. So could this impact their motor ability? The problem is, we don't really understand which parts of the brain are involved when we use motor imagery itself. So researching this is important, not only for us to understand how motor imagery difficulties could impact motor development, but also how we can use and target motor imagery in therapy to help improve people's movement. So we set out to study whether two parts of the brain that are important for movement are also involved in motor imagery in adults with and without DCD. These are called the primary motor cortex and the supplementary motor area. During 2 separate sessions, we used a technique called magnetic brain stimulation to temporarily reduce brain activity in these areas. We then looked at how this impacted performance on a motor imagery task. The task is simple. People have to determine if hand images rotated at different angles is a picture of a left or right hand. They usually make this decision by imagining themselves rotating their own hand in their mind to match the picture. We then compared performance from these sessions to a third session where no brain activity was reduced. Preliminary results show around 60% of participants with and without DCD had worse motor imagery when reducing activity in the primary motor cortex. We also saw this in around 70% of participants when reducing activity in the supplementary motor area. This tells us that both brain areas may be involved in motor imagery. But more importantly, people who have difficulties imagining movement, like those with DCD, may have problems in the functioning of these brain areas, and perhaps this could be the starting point to help us better understand and treat movement disorders.